Good day everyone, welcome to another lecture to Hematology MLS 413. I am Winbet Corpus and I will be your lecturer for today. We will be discussing the part 2 of the leukocyte development, kinetics, and functions of the leukocyte series, in which in this discussion we will be concentrating on the agranulocyte group. For the objectives of this discussion, we'll be able to describe the development, the function, the kinetics of the following cells: the monocytes and the lymphocytes, which comprises the agranulocytes. Basically, the monocyte maturation and development is just similar with your neutrophils, in which they come from the granulocyte monocyte progenitor cells, in which the monocyte colony stimulating factor is the major cytokine responsible for its growth and development of the monocyte. As you can see in the image on the right side, it begins from the monoblast to your most right, then to the promonocyte, and then to a monocyte, which is the mature one. So we'll be discussing each cells from the monoblast to the promonocyte and then to the monocyte. As you can see in the image at your left side, it begins in the stem cells, which is a multipotent hematopoietic stem cells, which will eventually respond to the cytokines and stimulus to produce the progenitor cells, in which they are the common mild progenitor, and then it will produce the GMP or the granulocyte monocyte progenitor cells, and will produce precursor cells in a form of monoblast, then to a promonocyte, and then we can see in the peripheral blood smear is the monocyte, and then in our tissues will be the macrophage. This would be the microscopic image from letter A, B, C, and D. So in your right side, this is the monocyte maturation diagram, in which it shows the characteristics of the nucleus, the nuclear structure, the chromatin, the vacuoles, and also the cytoplasm and the NC ratio, in which we have here the monoblast, and then the promonocyte and the monocyte. So shown here in the image that the nucleus shape goes from round to oval to an irregular indented to kidney shape or deeply indented shape as the cells mature. And then they also have multiple distinct nucleoli, which will become less distinct to not present as the cell matures. Also, the nuclear chromatin starts as fine or immature to fine spongy to dark blue, which is a mature form. And also the cytoplasm color goes from bluish to blue-gray to a gray-clear color. And vacuoles may be present also at all stages. As you can see in the image, from the monoblast to monocyte, they contain circular whitish structure, which corresponds to your vacuoles. Also, the cytoplasmic ratio increases. Now, let's begin our discussion with monoblast, in which it usually measures 12 to 20 micrometers, and its nucleus is characterized by round with folding or cleft with 1 to 2 nucleoli, while the cytoplasm is basophilic and non-granular, since monocytes are agranulocytes. And also take note for the NC ratio, it's usually 4:1 to 3:1, and it's usually difficult to distinguish from myeloblasts based on morphology alone in the microscopy. So this is the image on the peripheral blood smear of a monoblast, in which, as you can see, it has high nuclear to cytoplasmic ratio. And take note of the nuclei, it is round to slightly indented nucleus with prominent nucleoli, diba? We've mentioned a while ago that it could have 1 to 2 nucleoli. And sometimes, we mentioned a while ago that the vacuoles may be present from the monoblast to monocyte series. Also shown in the image is the prominent vacuoles that can be present on the monoblast in the peripheral blood smear. It also has high nuclear to cytoplasmic ratio, which is again 4:1 to 3:1. And also the nucleus is slightly indented to round to oval. Next on the monocyte series, the promonocyte, which usually measures 14 to 18 micrometer, and in raw duct it measures 12 to 18 micrometer. So the nucleus is also oval with slightly indented, folded, with less prominent nucleoli, at least 1 nucleolus, as compared to your monoblast, which comprises 1 to 2 nucleoli. So the chromatin is delicate, as shown in the image. And then the cytoplasm now becomes blue-gray from the basophilic agranular of the monoblast. And then the NC ratio is 3:1 to 2:1. So the right image shows the promonocyte, in which it has high nuclear to cytoplasmic ratio, indented nucleus with immature chromatin, which is delicate, then presence of vacuoles and less prominent nucleoli than the monoblast. So this is a very good representation of a promonocyte. According to Roddick, the pool of promonocyte is approximately 6 times 10 raised to 8 cells per kilogram, in which the production rate is 7 times 10 raised to 6 monocyte per kilogram per hour. And then they said that the promonocyte undergo 2 mitotic division in 60 hours to produce 4 monocytes. In cases that there is an increase in demand of monocytes, the promonocyte undergo 4 division to create or produce 16 monocytes in 60 hours. Also take note of this, that promonocyte has no storage pool of mature monocyte in the bone marrow, unlike in the neutrophils that it has a storage pool in the bone marrow. So when the promonocyte are produced, they are immediately released into the circulation. And then recent studies have shown that the promonocyte or the monocyte has a reservoir on the subcapsular red pulp on the spleen, and it usually responds to myocardial infarction or any disease. So this shows the peripheral blood smear appearance of a promonocyte and the peripheral blood smear. So it has high NC ratio, same with your monoblast. And then it has a more indented nucleus with immature chromatin, and then vacuoles may be present. And also there is less prominence of the nucleoli as compared to your monoblast. So this slide shows the difference between the monoblast and the promonocyte. As you can see, the indentation is slightly more prominent to the promonocyte when compared to your monoblast. And then they both contain your vacuoles. So when the promonocyte matures into a monocyte, it usually measures 15 to 20 micrometer or 14 to 20 micrometer. And it is deemed to be the largest cell seen in the peripheral blood smear. So what is the characteristic nucleus of a monocyte? Basically, it's round, or it can assume a kidney shape and may be folded with brain-like convolutions and absence of nucleoli. And then the chromatin is characterized to be loose and has lace-like or stringy appearance. For the cytoplasm, we have the monoblast, which is basophilic. We have the promonocyte, which has blue-gray. And for the monocyte, it has blue-gray cytoplasm with fine azurophilic granules, or also called the azure dust, which is a characteristic appearance of ground glass or frosted appearance. The cell membrane may have or contain blebs or pseudopods, since our monocytes is a macrophage or a phagocyte, which is used to eat up or ingest foreign organisms. So for the NC ratio, it has 2:1 to 1:1 NC ratio. So as you can see in the right image, you can see that the monocyte is kidney-shaped nucleus with mature chromatin. Vacuoles are commonly noted, and it has low nuclear cytoplasmic ratio when compared to your monoblast and promonocyte. So the monocyte basically comprises of 2 to 11% in the peripheral blood smear with an absolute count of 1.3 times 10 raised to 9 per liter. So the monocyte are slightly immature when they are produced from the bone marrow, and then eventually they go and mature to the tissues to become macrophages. So for the lifespan, the monocyte approximately lives or stays approximately 3 days, and for the macrophages, it usually varies depending on the location. So for the Kupffer cells, as mentioned in the book, it lasts for 21 days. Here is a typical microscopic appearance under the peripheral blood smear of a macrophage, in which it has horseshoe shape or kidney shape appearance of the nucleus with vacuoles, and it is larger or the largest cell in the PBS. So this is just the image on the peripheral blood smear of a monocyte. So characteristic horseshoe shape or kidney shape, and as you can see, it is larger than your neutrophil and also larger with your red blood cells and platelets. So on the bottom part, this is the appearance of the monoblast, the promonocyte, and the monocyte under the peripheral blood smear. So as you can see, the nucleus changes its shape from an oval or round to a more indented, and also the vacuoles increases. And as you can see, the presence of the blebs or the pseudopods on the cell membrane. As mentioned a while ago, that monocyte eventually goes to the tissues and become macrophages or histiocytes. So they migrate to tissues to become macrophages and become the tissue component of the monocyte system. They usually arise from immigrated blood monocytes and larger than monocytes. So macrophages are generally larger and usually measures 15 to 80 micrometer, and it contains irregular cell membrane with blebs and pseudopodia. So it is like an arm-like structure similar to your amoeba. So it has high nuclear cytoplasmic ratio and has oblong or indented nucleus. So I know that in histology you are taught that in each tissue component where the macrophages reside or stays, they have its characteristic name. So I'm expecting you to memorize this one from blood up to the intestine, the names of the monocyte or the macrophages. So this table is lifted from your RODAC. So this would be the monocyte destination, and then the different names in the different locations in the body. So we have liver, Kupffer cells. In the lungs, we have alveolar or dust cells. In the brain, we have the microglia, Langerhans cell for the skin, splenic macrophages seen in the sinusoids in the spleen, intestinal macrophages in the intestine. Then we have peritoneal macrophages in the cavities in our body. We have osteoclasts for the bone. We have type A cells for the synovial macrophages. We have renal macrophages or the mesangial cells in the kidneys. We have also reproductive organ macrophages. And the most potent and the most important is the dendritic cell, which is the most potent antigen-presenting cell in the lymph nodes. Now let's go to the functions of our monocytes or our macrophages. So they are defense against microorganisms. Specifically, we have the Mycobacterium tuberculosis, which is intracellular microorganism. We have fungi, bacteria, protozoa, and some viruses. They are also responsible for the destruction of aged or senescent red blood cells, denatured plasma proteins and lipids for recycling. And then as mentioned before, that they have this enzyme heme oxidase, which is responsible for recycling hemoglobin from the aged red blood cells. And also they are responsible for the antigen-induced blast transformation of lymphocyte. As we all know, diba? This was discussed in your immunology by Dr. Salaver that our macrophages plays a role in the transformation of lymphocyte. For example, they secrete interleukin-1 or what we call the lymphokine activating factor or the LAF, which help activate the T helper cells for transformation. And then they also serve as antigen-presenting cells, so they physically present and deliver the antigen to the specific membrane receptors to the other cells. So again, they are for defense, destruction, and then they have heme oxidase activity, and then antigen-induced blast transformation of lymphocytes. Based on Roddak, our book, he categorized the functions of monocyte by innate immunity function, adaptive immunity function, and housekeeping function. So let's discuss first the innate immunity function. So they have toll-like receptors, which is used for the recognition of bacterial pathogens, which would eventually lead to the inflammatory cytokine production and eventually phagocytosis. They also have this substance called the NO or the nitric oxide, which is cytotoxic to viruses, bacteria, fungi, protozoa, helminth, and tumor cells. And also they have Fc and complement receptors. So what's the use of this? So diba, macrophages or monocyte are phagocyte. So diba, if there is masyadong malaki yung bacteria for it to phagocytose, so yung mga opsonins like your C3b will coat the bacteria. For example, in this case, the red gram-negative bacilli there. So they will be coated by C3b, and that C3b will be recognized by the complement receptor present in our macrophages, so that it would be very easy for it to engulf or phagocytose. Now, for adaptive immunity function, so the macrophages, for example, the dendritic cell, will degrade and present the antigen fragment. So they serve as antigen-presenting cell. So just a fun fact that the dendritic cell is the most efficient and most potent antigen-presenting cell. Always remember that. This came out on our review sessions during our medtech review days. So they interact also with the T and B cells for the production of adaptive immune response. So for the housekeeping function, so from its word housekeeping, so they remove debris and dead cells at the site of infection. For example, you have infected wound, and then magtaka na lang kayo bakit nawala na yung mga inflammation doon or any mga marumi doon, because yung mga patay na cells doon kina-remove ng monocyte or macrophages. So they also remove or degrade aged red blood cells or the senescent red blood cells, and then they maintain the storage pool of iron and proteins used in synthesis of coagulation factors, complement interleukin, growth factors, and enzymes. So always remember that our monocyte has innate and adaptive immunity function and housekeeping functions. Now let's go to another type of cell, which is our lymphocyte. So we know that we have several types of lymphocyte. We have T cells or T lymphocyte, we have B lymphocyte, and then we have the NK or null cells. So this slide is very familiar to you since I discussed it already. So the T cells are the most abundant, comprising 60 to 80% of the population of the lymphocyte. They are long-lived for 10 to 10 years. And then they are responsible for the cell-mediated immunity as discussed in serology and immunology. And then we have the B cells or B lymphocyte, which is the second most abundant, comprising 10 to 20%. They are short-lived, 3 to 4 days only. And then they are responsible for humoral-mediated immunity. And then we have the NK or the null cells, which comprise 10%, which is responsible for the tumor host defense. So this is used for the immunosurveillance in the body. So if ever may mga cancer cells, they are the one who will first respond to it. So now let's talk about the features of our lymphocytes. So in the peripheral circulation, they comprise 18 to 42% of the population of the white blood cells. So they have the absolute count of 0.8 to 4.8 times 10 raised to 9 per liter of cells. And then they are called not end cells because the resting cells, when stimulated, will be undergoing mitosis, and then this will produce two types of cells. We have the memory cells, which has memory na matandaan nila yung mga na-encounter na nila ng mga antigens noon, and then we have the effector cells, or sila yung mga movers. And then they recirculate from the blood, then to the tissues, and again to the blood. So we have rearrangement of antigen receptor genes seen in the B and T cells. What's the purpose of this? This is to produce variety of antibodies and surface receptors. Diba, the function of lymphocyte is very important because they are for the immune response. So meron silang mga memory na para second time around na i-attack tayo ng certain microorganism, mas mabilis yung ating response with regards to it. We have also the T and NK cells which develop and mature outside the bone marrow. For example, our T cells, diba, they are from the bone marrow. The T and the B cells are from the bone marrow. But our T cells does not mature in the bone marrow. They mature in the thymus. So we have already discussed the process on what's the negative and the positive selection for T cells. And then we have the B cells, diba, they are from the bone marrow, and then they mature into the bone marrow also, and then they undergo central and peripheral tolerance, something like that. So if we have blast and to site in our monocyte, we have also that in your lymphocyte. From our hematocytoblast or your hematopoietic stem cells, and we have the lymphoid stem cells, and from the lymphoblast, it will produce a prolymphocyte, and then into mature small lymphocyte or medium lymphocyte or to a large lymphocyte, and eventually when it encounters an antigen, it will produce a plasma cell and eventually producing your antibodies. So let's talk about the first stage, which is the lymphoblast, which is immature. Then it contains 10 to 18 micrometer nucleus, or I mean the size of the cell is 10 to 18 micrometer, with a nucleus that has coarse chromatin that is round to oval with 1 to 2 nucleoli. So the cytoplasm basically has no granules since lymphocyte belongs to a granulocyte series. They have smooth cytoplasm with moderate to dark blue cytoplasm. Next, we have the prolymphocyte, which is same with the with our lymphoblast, which is 10 to 18 micrometer, but some are smaller. The nucleus now in here is more clump chromatin than round to oval with 1 to 2 nucleoli. So the granules again has no granules. The same with your lymphoblast, smooth with moderate to dark blue cytoplasm. As you can see in the right image, you can see the 2 nucleoli present in the nucleus of the prolymphocyte. Now we have the mature small lymphocyte, which measures 8 to 10 micrometer, the same size with the red blood cells, diba? If wala doon sa choices yung what is the histologic ruler under the peripheral blood smear, if walang erythrocyte, you choose the small lymphocyte. So the nucleus now is dense chromatin when compared to your other type of cells, or in this series is they have more clump chromatin, and then they have round to oval nucleus now without nucleoli. So the cytoplasm is basically easy to remember. We have the robin's egg blue appearance of the cytoplasm, in which they have thin rim around the nucleus. So as you can see here in the image, ganyan daw yung color ng cytoplasm niya. It's robin's egg blue. So as you can see in the peripheral blood smear, it only contains a thin rim of cytoplasm surrounding the nucleus. So we mentioned about the mature small lymphocyte. So we also have medium lymphocyte, which measures 10 to 12 micrometer. So the nucleus is not as dense chromatin when compared to your small round to oval small lymphocyte. So again, the cytoplasm now is more abundant since medium lymphocyte naman siya. And then the cytoplasm is pale to moderate blue. So later on, we'll have a slide that compares the small, the medium, and the large lymphocyte. Now let's proceed to the mature large lymphocyte, which measures 12 to 16 micrometer with round to oval nucleus without nucleoli. And now the cytoplasm is now more abundant when compared to your small and medium-sized lymphocyte with clear to very pale blue cytoplasm. So as you can see here, mas marami na yung cytoplasm niya when we compare it to your small and medium lymphocyte. So yung small, diba, thin rim lang. And then yung medium natin is meron siya pero konti lang when compared to your large lymphocyte. So this slide is very good because this depicts the small and the medium and large lymphocyte. As you can see, the small lymphocyte only thin rim. The medium is medyo marami lang siya konti. And then the large is very large siya talaga. Very abundant cytoplasm na siya. So, dito hindi masyado ma-appreciate yung robin's egg blue na cytoplasm because hindi masyado siguro maganda yung stain nila. We also have a special subtype of the lymphocyte, which is the plasma cells, in which meron din siyang plasmablast, proplasmacyte, then plasmacyte, or the plasma cell, which is the mature stage. So, we have the plasmablast, which measures 18 to 25 micrometer. The nucleus is eccentric, and the cytoplasm is basophilic, abundant, and granular. We also have your proplasmacyte, which measures 15 to 25, that has eccentric again, nucleus, and then we have densely basophilic, agranular. And then we have the plasmacyte or the plasma cell, which measures 8 to 20 micrometer, which has eccentric, characteristic cartwheel pattern of nucleus. And then we have cytoplasm of deep basophilic, moderately abundant, agranular. They term as large HOF with perinuclear halo. So later on, I will be showing you what the large HOF perinuclear halo looks like. So this is the appearance of your proplasmacyte, which has eccentric nucleus with intensely basophilic agranular cytoplasm. This comprises 15 to 25% of your lymphocytes. Now we go to your plasma cells, or what we call the plasmacyte. Again, emphasis on the eccentric cartwheel pattern appearance of the nucleus with large Hopf or perinuclear halo. As you can see in the image, yan, di ba, may nucleus siya, and then beside it is your prominent perinuclear halo or your Hopf, or means clearing lang siya. Parang distinct structure siya compared to your cytoplasm. So, madali lang siya matandaan or makita sa peripheral blood smear. So, nakita nyo dito sa peripheral blood smear at your rightmost at the top picture. So, nakita nyo dyan, meron siyang basophilic cytoplasm, and then you can see the somewhat may clearing zones between the cytoplasm and then into the nucleus. So, yan yung tinatawag nating large Hopf or your perinuclear halo, which is unique for your plasma cells or your plasmacytes. So another characteristic of your plasma cell is they have eccentric nucleus with spoke wheel or your cartwheel appearance of the chromatin, or it has clock-like chromatin. So tandaan yun because it came out in our board exam. We have eccentric nucleus with spoke wheel appearance of chromatin. So yun pa lang, maalam nyo na that is your plasma cell. So the function of your plasma cell is to produce antibodies. So that ends our discussion about your agranulocytes. So the main reference of this lecture is from Rodak, page 127 to 132. And then I have a good atlas, can be seen on your internet. So you can browse it, and then you can see a lot of pictures there of your cells. So you can practice identifying them. So I hope you study well and do well on your examinations, and I hope you will pass Hematology 1. Thank you, everyone.